and I'm excited to get to talk to you about some of the research that I've been doing for the last few years. Um, so let's see. I'd like to begin. This is probably not news for most of you, but many bees are in decline. Uh, Bumblebees in particular are my maybe second favorite bee taxa, and they're in trouble across the globe due to a number of different stressors. So there's this figure here shows what we think of as kind of the big five of things that are driving bee population declines. And so we can think of um, a lot of these obviously are generated by human activity. Agriculture being one of the large drivers there. So we see uh, pesticide exposure um, as being a large contributor to declines. Um, Poor nutrition brought about through the loss of flowers due to the expansion of industrial agriculture and urbanization. Um, and that loss of habitat also leads to a loss of places to nest. So habitat for bees isn't just the flowers that they need, but also the, the bare ground or the cavities in um, plants and rocks and so on that they use to nest. And then certainly climate change is becoming an increasingly significant issue, leading to mismatches between when the flowers are blooming and when the bees are flying, um, and leading to acute stress from too much heat for many of these bees. And then finally, the stressor that I'm going to be talking about mostly tonight is pathogens. So disease is implicated in the decline of a number of our most iconic bee species. And in some ways, pathogens and disease are different in kind from these other stressors because pathogens are an essential part of any ecosystem. And in fact, they often help to maintain diversity by making it so that more dominant species are kept in check because they are exposed to more disease. And so, in some sense, it's an odd thing to be talking about pathogens as a problem for these species because, again, they're an entirely natural part of the ecosystem. But the thing is that what we've seen is many of our rarest bee species are actually at the same time the ones that are most afflicted by um, pathogen loads and pathogen exposure, which suggests that in this case, these pathogens are not helping to maintain diversity, they're actually driving it down. And that's probably not due to the effect of the diseases on their own, but more their interaction with some of these other things, like say pesticides or lack of nutrition. And so I think from coming from an ecological background, I also am most happy to study pathogens because they are this really cool system to think about how bees are responding to complex interactions with other species, including ones that we often tend to ignore or actively repress. And so in the study of bees and their response to disease, there's been a lot of recent interest in thinking about how those effects might be combated by the bees themselves in terms of what they eat. And so this is data from a recent review that I collaborated on looking at evidence for medicinal effects of various compounds produced by plants. Um, and we found 51 different chemical compounds that can be found in pollen or nectar or resin that have known medicinal effects for bees against one or more diseases or um, like, to mitigate effects of pesticides and so on. And these are really widespread. So at least 147 plant species produce one or more of these chemicals, including the ones shown here, like sunflower and coffee. And these figures show just the evolutionary relationship among plant species. And so you can really see that there is they're really distributed widely across the plant kingdom. And it's not just bees, obviously, that are getting medicine from plants. Many of our most iconic human medicines also have their origin in plants, even if now we may be synthesizing them without the use of those plants. But humans have used plants for probably millennia to combat diseases of our own. Which raises the question, perhaps, why do plants provide such a rich source of medicine? It's probably not for out of the goodness of their heart, but rather due to the fact that plants, they can't run, and yet they have a lot of things that are very happy eating them. And so they have to come up with other approaches to dealing with those enemies. And one of the most effective approaches that they've come up with is chemistry. So plants are wizards at producing all sorts of chemical compounds, many of which, these are not helping the plant grow any better in the absence of predators, but really it's defending themselves from predators. And those range from things like structural compounds that make it so a plant is tough and fibrous, to these more medicinal compounds that are actually toxic at high doses, but for humans, we're not insects, and so we're not quite as affected by them as the intended recipient. But so, given that it's actually a bit of a puzzle that we find all of these chemicals in pollen and nectar, right? So pollen and nectar we think of as rewards for bees. And so why would plants be sticking these toxic compounds into pollen and nectar when it seems that their intention is to deter from eating them? I should maybe clarify this a little bit. So this is actually it makes a bit more sense that plants would protect pollen. So again, when bees are visiting flowers, they're looking for two things. They're looking for pollen, which is their primary source of protein and lipids, and they're looking for nectar. And nectar is really exclusively for the bees to feed on, or other pollinators. The plants have no other reason to produce it other than to attract pollinators. The pollen, on the other hand, this is both food for pollinators and the male gametes of the plant. So in some sense, it makes sense that pollen might be rewarded. They don't want bees to just be scarfing down all of that pollen. They want most of it to go to other flowers so they can reproduce. But nectar, it's really odd. You would think that plants would be sticking these presumably toxic compounds into this attractive sugary liquid. And so one possibility is that these plants can't help themselves. Like they're producing these chemicals to defend their leaves and their roots and their stems, and some of it just winds up in nectar by accident. And it's better to defend your leaves than have everything get eaten, even if it means you deter a few beneficial pollinators. But the other possibility, and the one that I've been thinking about a lot recently, is maybe it's actually good for the plants to put these compounds into their nectar. And so how would that work? Well, there's growing evidence that these toxic compounds might actually attract pollinators. So the best evidence we have for this is with caffeine. So there have been a number of studies showing that caffeinated flowers are going to attract more bees, seemingly because it makes those bees remember the flowers better and more likely to come back, more likely they're a social bee to tell their nestmates to go out and find those same flowers. So even though these compounds may be toxic in high doses, at low doses, just like us, bees perform tasks more efficiently with a little bit of caffeine. Another possibility, sort of non-exclusive to that, is sort of has its basis in the observation that disease can alter foraging preferences. So certainly this is true for me. I know when I get sick, I tend to prefer different foods than I am craving when I'm feeling good. And it's not true just for humans. Plenty of animals also change their feeding behaviors when they're sick, and in some cases do so to actively reduce that infection, meaning they're self-medicating. So some of the earliest evidence of this was from chimpanzees, where they'll go out and chew on these really bitter leaves and really fibrous leaves when they are sick with an intestinal parasite. And it seems that these leaves actually kind of scrape out the intestinal walls and reduce the parasite infection. Um, and this is a plant that when the chimpanzees are healthy, they will avoid, but they are self-medicating when they're feeling crummy. And it may be one thing to have a, you know, highly intelligent vertebrate doing this, but we have also found evidence in a few different insects of self-medication. So primarily the strongest evidence is from Lepidoptera. So here, monarch butterfly caterpillar, and as I imagine most of you know, monarch butterflies, uh, the caterpillars feed exclusively on milkweed. Milkweed has these quite toxic cardenolides that wind up in um, the bodies of these monarchs and allow them to be distasteful and toxic to things that eat them. So it deters predation. But at high concentrations, these cardenolides do also have negative effects on the monarch butterfly. They're only so good at sequestering them in their tissues. And at high doses, they will stunt their growth or even kill the caterpillars. And yet, when monarch butterfly females are infected with a parasite, they will preferentially choose to lay their eggs on milkweed that have particularly high levels of these toxic cardenolides. So these are plants that are bad for growing caterpillars because they stunt their growth, except when those growing caterpillars are infected with this vertically transmitted parasite that gets passed from mother to offspring, in which case the, the bad effect of that high cardenolide concentration on growing caterpillars is counteracted by its even worse effect on the parasite, where it actually tends to clear the parasite infection and allow these caterpillars that might otherwise die from parasitism to survive, even if they grow a little bit slower because of dealing with all these toxic compounds. So there's, again, growing evidence for a diversity of medicinal behaviors in animals from across the animal kingdom, including insects. And in a lot of ways, bees would seem to be a poster child for self-medicating behavior. Because so as I've already shown you, we know that their food sources are rich in these plant secondary metabolites, many of which have antiparasitic compounds. We know that bees, like all organisms, are infected by a variety of diseases. And even more, we know that bees are really sophisticated at determining the nutritional composition of pollen and nectar and making choices about what they're eating based on, the pollen to or the protein to lipid ratio of plants, for example, or the sugar content of nectar. And yet, there's to date been very little conclusive evidence that bees are doing any kind of self-medication with this, potential pharmacopoeia of flowers out there. I'd say the one sort of asterisk to that is from honeybees, where there is fairly strong evidence that their use of propolis to seal the hive structure is influenced by disease, and so they'll use particular plants to create that propolis in the presence of parasites that tend to help with parasitism. But that's kind of a different kind. That's not really them feeding, and that is not this kind of individual self-medication. It's more of a colony-based situation. So given all that, I've been setting out to figure out if we can detect self-medication in bumblebees. So thinking about whether sick bees are preferring antiparasitic nectar, and then also thinking about, does that have consequences for plants? We know that bee pollination is essential for the reproduction of many plants. And so could this mean that medicinal plants have, say, an advantage or a disadvantage depending on how widespread disease is in a bee population? To look at this question, I've been working with a really fun system. I'll just introduce you briefly to the players here. So the bee that we're working with is very familiar Bombus impatiens, common eastern bumblebee. I haven't spent much time in Maryland, but I suspect this is probably one of the more common mid to late summer bumblebees, particularly in more disturbed habitats that you'll see. Certainly in Toronto, in the August and September, there's just gazillions of them around. And not surprisingly, given their abundance, they're a really important pollinator. So they're one of relatively few bumblebee species that have been sort of commercialized. So they're bred in captivity and used for pollinating greenhouse crops like tomatoes and so on. Which is really nice for me because it means we can also maintain them in the lab and do manipulative experiments with them in a way that is really difficult in the wild. These bees are host to lots of different parasites, but the one that I am working on primarily is called Crithidia bombi. So this is trypanosomatid that's related to leishmaniasis and Chagas disease, the two tropical human diseases. This one has a simpler life cycle than those which have sort of multiple hosts. Crithidia just completes its whole life cycle in a bee by latching onto the hindgut and extracting nutrients from the lining of the intestinal wall. Then after a while, it's reproducing, new cells get shed when the bee poops, and bees aren't the most sanitary, so they are frequently pooping where they eat. And then other bees will come along and in sipping up some nectar, will also sip up some Crithidia, and they get infected that way. And also with social bees like bumblebees, they'll bring it back to their colony and very quickly can infect the whole colony. So this is a highly prevalent disease in most places that we've looked for it. So in the northeastern US, probably over half of Bombus impatiens are infected with it at the height of their foraging season. And that can range up to like 80–90% of bees in a population. With that high prevalence, you can imagine it is not usually lethal. If it were, we would not have so many Bombus impatiens flying around. But it does have negative effects, particularly for bees that are otherwise stressed for nutrition or other diseases. And so the most significant effect is that it reduces the queen's overwintering survival, but it also reduces foraging efficiency and leads to sort of slower growth of colonies. So significant, but not deadly parasite. And then the plant that we've been working with is Monarda fistulosa, or wild bergamot. So this may well be familiar to many of you. It's common across Eastern North America, going into the Rockies, more or less. And the thing that's really cool about Monarda, that is the reason that I focused on it, other than it being super attractive to bumblebees, is that it has what we call chemotypes. So these are multiple genetically determined chemical phenotypes that occur together in a population. So any given plant will produce one chemical compound in high abundance, and it produces that same compound throughout its life, but it can breed with other plants that produce a different chemical compound, and they can have offspring that will have a sort of mix of these different chemotypes. And so you can go out into a field of Monarda fistulosa like this, and if you rub your fingers on the leaves or sniff the flowers, you'll see that most of them have a sort of herbaceous smell, kind of like thyme or oregano. And that's from these two chemical compounds, thymol and carvacrol. So I would say in most populations, probably 90% or more of the individual plants have this thyme-like smell. But if you're lucky, you'll find others that smell like lavender or smell like lemon or even eucalyptus, all growing together in the same field. And for a long time, this was thought to be actually vanishingly rare. So in the 70s, people up in Wood Buffalo National Park in northern Canada came across a couple of these geraniol producing plants, ones that smell like lemon, uh, and thought it was just this sort of, fluke mutation leading to this one population. But then in the early 2000s, Ken Kiefover-Ring was doing his PhD research in Colorado and found a couple more plants in the Southern Rockies that had uh, one of these other compounds, one that smells like lavender. And then in a pandemic, we had a lot of time to go out and smell Monarda plants. And so we've found quite a few more populations. So Ken also found these ones in Madison, Wisconsin. And then I was finishing up my PhD in Ann Arbor, Michigan, and found all of the same compounds there. And then have since moved on to Toronto, and we found similar compounds there. So it seems like this isn't actually just sort of a fluke. These keep showing up in populations. And seem to persist pretty stably over time. So Ken has gone back to that population in southern Colorado we initially found in 2006, and they're still there at about the same density in the population now, nearly 20 years later. And just a cool aside here, I think we're thinking this is this cool new discovery that we've made, and then there's this ethnobotany book that came out in 1992 that points out that indigenous peoples in the Great Plains had 4 different varieties of Monarda fistulosa that they recognized. I don't know for sure. Like, I think in this book they're thinking that these are different subspecies, but I would bet that what these different names refer to are actually the different chemotypes, particularly because these names were associated with different medicinal practices that these indigenous peoples used, which just yet again points out how, here we are, Western science, thinking we are so advanced in our knowledge and often can learn a lot from people who have been on the land longer and had ways of thinking about and knowing the landscape that we're still struggling to keep up with. So the cool thing, the first thing that I found out with this plant that got me really excited about this is when you feed bees that are infected with this Cryphidia parasite nectar from these different chemotypes, some of the chemotypes have nectar that is antiparasitic, meaning bees feed on it and then afterwards they have less Cryphidia. And some of these compounds are not antiparasitic, so they don't have any effect. So specifically, the really common ones, thymol and carvacrol, both are medicinal, also eucalyptol. And then these non-medicinal ones are the ones that smell like lavender and lemon. And so this sets up this really nice system to ask whether bees are self-medicating, because the only thing that's different about the plants is their chemistry. So we can say for sure that if bees are choosing one over the other, it's not because one is more nutritious or more beautiful, but because of the chemistry. And so if bees are self-medicating, we'd expect that healthy bees should not really discriminate among chemotypes, or maybe even they would dislike the medicinal ones because maybe they're bitter tasting. If that's the case, then we would expect these plants to be equally well pollinated, regardless of whether bees are healthy or sick, and therefore produce similar numbers of seeds. By contrast, if the bees are infected with Cryphidia and they're self-medicating, we'd expect them to visit these medicinal plants more, leading to better pollination and more seeds for those plants relative to the non-medicinal ones. And so to test this hypothesis, we did a bunch of experiments in tents, look like this picture here. So in each tent, we had a couple plants. It was either a comparison between one medicinal and one non-medicinal plant, or two chemically dissimilar medicinal plants to kind of see whether, maybe it's just the taste of time that they don't like rather than the medicinal versus non-medicinal aspect. Into these tents, we introduced bees that we uniquely marked with a different color pen so we could follow them individually. So you can see the one in the photo here has a little white dot on its back. And we knew whether these bees had the disease or not. And then we just followed them, saw what they did, and then later on looked at the pollen that they left behind on the flowers and how well the flowers produced seeds. And what did we find? Well, the first thing that was super exciting is that these bumblebees do seem to be self-medicating. So consistent with our hypothesis, you can see in this picture, this graph showing you that on the left, we have our bees that are not infected with the disease, and on the right, the infected bees. In blue are the non-medicinal plants, and in orangey red, the medicinal ones. And basically, you can see that when bees are healthy, they're having an equal likelihood of visiting a flower regardless of its chemistry. But with these infected bees, we have them much more likely to visit the medicinal plants and less likely to visit the non-medicinal ones. And then when we looked at these comparisons where we had two medicinal plants, we were happy to see that there wasn't any sign of an influence of infection on how they were foraging, suggesting that this preference for the medicinal chemotypes is due to their medicinalness rather than to just the flavor of the compound. So that was sort of our first big exciting discovery. But then, I'm really interested in what this means for pollination. And so we next wanted to look at how well the bees are pollinating these different chemotypes depending on whether they're sick or healthy. And here again, we find evidence for the effects of self-medication on not just behavior, but then on pollination. So here I'm showing you the number of pollen grains that were left behind after a bee visited a flower. Again, depending on whether the bees were uninfected or infected. And in this time, you can see the same pattern, more or less, where infection meant that pollination improved for the medicinal plants, but was worse for the non-medicinal plants. So very cool stuff. Then we were kind of bummed to wait for a couple more weeks and collect these flower heads once they had produced seeds and find that we had no effect of infection on which chemotype did better. We see that infection tends to make it so that plants produce fewer seeds, which maybe makes sense given what I've told you about how infection reduces their ability to forage. Although this is in itself a cool finding because we haven't been able to document before that infection actually has a negative impact on the plants that are being pollinated. But still, it was kind of disappointing to not see a difference between the chemotypes. But then we're kind of looking at the data and we realized, what? If I show you this graph of the pollen deposition, this is like not a lot of pollen. These plants are receiving on average maybe like 3 grains of pollen. And that's probably not enough to actually allow them to reproduce. They're probably limited in their reproduction by the amount of pollen that they're getting. And this was confirmed because those plants that were inside of our tents produced way fewer fewer seeds, than the flowers that were open to pollinators all the time. Meaning these poor plants, they were doing the best they could, but they couldn't really produce seeds the way that they wanted to. And so maybe the lack of an effective self-medication on their reproduction is just kind of an experimental fluke. So then we went back the next year and set up a new experiment, also intense, but now we put in more more plants in each tent, so there was more opportunity for cross-pollination. Bees had 41 more hours to forage than in the first experiment. And this time, the results did seem to be, well, at least more interesting. So here I'm showing you how many seeds each flower had produced. And here again, there's like maybe a hint of those medicinal plants in orange producing more seeds when they're relative to the non-medicinal plants when they're infected. But even more, we see this big drop in the number of seeds that are produced. So again, bees seems like sick bees are not so good at pollination. Now I'm going to show you if we scale up. So instead of looking at how many seeds are produced by each flower head, we're going to look at the whole plant. And here we see more of a hint of self-medication happening, right? So here we see that the seed production in the non-medicinal linalool plants declines pretty steeply as we go from uninfected to infected bees. Still, those of you who are like versed in statistics, a p-value of 0.07 is like, not exactly what you want to see. But maybe there's something going on there. So then to dig into this a little bit more, see if there's something else going on here, we took those seeds that we had collected and we planted them in the greenhouse, let them germinate, and looked at the germination rate. So did that differ between plants that were pollinated by infected and uninfected bees? And then we looked for each of those offspring what their chemotype was, so we could get a sense for, imagine we have a population of two plants, one medicinal and one non-medicinal. What's the next generation of that hypothetical population of two plants going to look like with uninfected bees versus with infected bees? And I think the results are pretty striking. So here's what that first generation will look like if we have uninfected bees. So interestingly, even with uninfected bees, the next generation has more medicinal than non-medicinal plants. And this has to do with the genetics of these different chemotypes. But so, the bulk of the offspring for either chemotype parent are the medicinal ones. But for those infected bees, we see a pretty strong and striking shift where we get not only more medicinal plants, but even more strikingly, we get way fewer non-medicinal plants in that next generation when those plants have been pollinated by infected bees. So in fact, we see like a 43% decline in the number of these non-medicinal plants in the next generation when they've been pollinated by infected bees versus uninfected bees. So it's a pretty striking result. I'll say, it was pretty cool, I have to say. And so it paints this picture of kind of a complicated little flow diagram. So I'll sort of walk us through it. So what it seems to be happening is that these parasites are negatively affecting bees, but they're also shifting their foraging preferences. So now they're preferring to feed on these medicinal plants at the cost of visiting these non-medicinal plants. And so they're actually having this indirect positive benefit of the parasite on the medicinal plants, even as the medicinal plants are having this direct negative effect on the parasite by literally killing them. And so this makes us wonder, is this part of the reason why we have these multiple chemotypes in the population? And to sort of walk you through this, imagine that we have a low level of the parasite in the population. At that point, the non-medicinal chemotypes would be faring better than the medicinal chemotypes. And so their relative abundance in the population would increase. That would mean, though, that there's less of this medicinal effect in the landscape, and that could allow the parasite population to increase, at which point now the bees are favoring these medicinal plants, which in turn would lead to a reduction in the parasite loads, which then would push the bees' preference back towards non-medicinal plants. And over time, we could see this sort of flip-flopping between bees' preference and consequently the fitness of these plants, sort of out of sync with the parasite prevalence. So kind of almost like a classic predator-prey cycles dynamic happening. Yeah, that's just summarizing that. So basically, I don't know if this is happening, but I think it's a cool story, and one that I'm interested in pursuing further. But I have to say, especially since this is an entomology crowd, that this isn't the whole story. So I've mostly been focusing on the bees and the parasites, but there are other insects that are out there among these plants, and in particular, uh, the summer that we did that second tent experiment, our plants that weren't in tents got attacked by Japanese beetle and oriental beetle. Um, and we found that those beetles had a strong preference for the non-medicinal plants. So perhaps not surprisingly, this thymol and carvacrol compounds that are more toxic for the parasite are also more toxic for the beetles. Um, and so in populations where we have both disease and bees and herbivores all hanging out together on these plants, the dynamics of how this would affect the fitness of the different chemotypes is probably more complicated than I'm letting on here. And so in this case, it would seem like this would actually reinforce the positive selection on medicinal plants, which again is kind of consistent with the facts on the ground that these medicinal chemotypes are usually far more abundant than the non-medicinal ones. So that's sort of the story that I wanted to tell you about this. I'm going to conclude with sort of some thoughts on where our research is going, and then maybe I think in the abstract I said I would talk a bit about what this means for gardeners. May leave more of that for the Q&A, but I'll touch on it briefly. So one thing that we're really interested in looking at is what's going on with wild bees. So like I said, this work so far has been done with bees that we keep in the lab and that we sort of coddle and have in this very nice environment and then bring them out to the field just for our foraging experiments. And then they're in these tents where they have a pretty constrained choice. They can't just fly around and feed on any flower that they want. And so in the wild, are these bees still making these choices for self-medication, especially when they have this whole cornucopia of other options for flowers that they could feed on? So we've started looking at this by sampling bees out in the wild, and you can, rather than have, so in the lab, we often will dissect the bees to look at their guts to screen for the parasite, but because it's transmitted fecal orally, you can also just let a bee poop in a vial out in the field and check the feces for Clyphidia and then let the bee go. So we're doing more of this non-lethal monitoring of the parasite to see whether out in the wild, sick bumblebees are also spending more time on these medicinal plants than the non-medicinal ones. And so the same goes for the plant. So, in the case where these plants are only pollinated by our bumblebees, we're seeing this signature of indirect selection by the parasite for the medicinal chemotype. But whether that's actually happening in the wild, particularly when these plants are exposed to all sorts of other, not just pollinators, but other kinds of herbivores and other things that interact with them, are we still going to find that the parasite seems to be sort of putting this evolutionary pressure on the plant? To be determined. And then we're also really interested more in looking at the dynamics within the bee. So like, what is it that is making these bees change their foraging behavior? So to do this, we've been doing some, just completed a project looking at the gene expression of infected and uninfected bees in their brains to see whether there's differences in their sensing patterns that might go along with their preference for these different compounds. But again, don't know the answer yet. And then, thinking often when I talk about this work, people, what they really want to know is like, okay, so are medicinal plants going to save the bees? And so the first thing that we need to do to answer that is see whether these effects, the positive effects on the individual bee scale up to influence fitness of the actual bee colony, so their ability to produce new queens and have those queens successfully overwinter. This is obviously a lot more challenging than having bees in a lab and feeding them just for a couple of weeks of their life. But it's something that we're working on, and something that there is not for this Monarda system, but there is evidence from some other sources that other plants are able to have a strong enough medicinal effect that they end up influencing the sort of fitness of the colony and potentially even the sort of population scale success. So for at least for this common eastern bumblebee that I'm talking about, sunflower pollen seems to be a really effective medicine against the Cryptidia bombi parasite. They don't seem to be actively choosing sunflower, but nevertheless, if sunflowers are in the landscape, bees are collecting it because it's a rich source of pollen for their nutritional purposes. And in the process, it's helping to rid them of parasites to the degree that it's in landscapes with high concentrations of sunflower, you see these uninfected colonies then producing more queens than colonies in landscapes with less sunflower. So I think I'd say there's some potential there for managing disease in a way that promotes pollinator health through the plants that we choose to plant, but it's probably premature to tell everyone to go out and plant medicinal Monarda. Although I'd say do plant Monarda, it's a great plant. Bees love it, other insects love it. So whether it is going to rid the bees of Cryptidia or not, it's a good choice to plant. Yeah, and then of course there's this question of beyond Bombus impatiens, beyond Monarda, are there other diseases that are afflicting bees that they're combating with self-medication? There's sort of a whole host of questions there that we're looking forward to answering, or at least puzzling over in the coming years. So with that, I'd like to thank you all for listening and thank my collaborators and particularly the undergrad students who have put many hours into watching bees and counting tens of thousands of seeds. And then my funding sources, and most of all, the bees and the plants that are the real partners in this work, and in many cases have given literally their lives to it. And yeah, with that, I'm happy to take any questions. Go ahead and stop sharing my screen.
All right. Very good. Very interesting. I have a few questions that I'd like to ask, but I want to give other people a chance first to ask questions. So, anybody, feel free to raise your hand or speak up and ask a question.
Well, I don't have a question. Hi. Um, my name is Tiffany. I'm one of Professor Kurtz's students. I don't really have a question. I just have a comment. It was very interesting for me, everything that you talked about the bees, because I myself am allergic to bees. So to hear them, the bees being sick and going to different plants and things, it was very interesting because I'm a severe case of being allergic to bee stings. I actually just instantly faint when I'm stung by bees. So it was just nice to learn something about them. Even though I'm very afraid, I try to stay clear of bees at all the time.
Yeah, thanks so much for that, Tiffany. I'm sorry to hear that you're allergic. I wonder, I think there's some specificity to bee toxins, so it may be that I don't know if you've been stung by a bumblebee, but often people who have strong reactions to honeybees, I'm not saying you should go out and try it, but aren't um, so affected by bumblebees.
Yes,
But yeah,
I have
they're doing
been.
good things, even if they are also causing problems for some of
Yes.
us.
Yeah, I've been stung by a bumblebee and the regular bees as well. Yeah.
Okay. Okay.
I have.
I wanted to ask a question, Gordon. When you
Yeah,
did
please.
your experiment in the tents, and you had bee balm, the two types of bee balms, right?
Hmm.
What number of bumblebees that were introduced to the tents? How do you control for that, or is that a factor necessarily?
Yeah, good question. Yeah, so we would typically put in just three. Well, so in the first year it was three bumblebees, and the next year it was five because we had a few more um, plants. And the reason for that number was basically bumblebees are really hungry and they will very quickly deplete all of the nectar in these plants. They don't produce a ton of nectar in each flower. And so, we wanted to get an accurate sense for whether the bees had a preference and if they were just starving and desperate for any nectar that was there because they already consumed it all, they wouldn't accurately show their preference because certainly their preference is some sugar over no sugar, even if that sugar tastes nasty. So that was the reason for that fairly small number.
Okay.
So
And
can I ask a question?
sure.
Okay, so thank you for your talk, Dr. Fitch. I would turn my camera on, but my camera broke, unfortunately. But for the parasites you were looking at, do you I guess the bumblebees pick them up mostly through their feces. They poop on the flower or whatnot. How long do those parasites last on the flower? And do you know if there's a difference? Do they last longer on the flowers with certain chemotypes than on?
Yeah, great question. So it depends. So in general, it depends on where on the flower they poop, and it also varies from flower species to flower species. So these parasites are they're pretty sensitive to both desiccation, but also to sunlight. And so Monarda, is kind of the bees kind of crawl inside the flower, and so often when they're pooping in the flower, they're pooping in an area that's fairly protected. And so on Monarda, the parasites can last a few hours, I would say. Um, and yeah, as to the question of whether the um, it matters which chemotype they are, we've looked at that a little bit, and I sort of thought that it would matter because, again, this compound isn't just in the nectar; it's also in the flowers have these little hairs, trichomes, and when they break, it exudes some of these compounds. And so we figured that maybe the parasites would be exposed to the chemical just sitting on the flower. We haven't found evidence for that, so it doesn't seem like the chemotype changes how long the parasite persists. Um, and just another interesting thing that we're thinking about that is because of that, if the sick bees are preferentially foraging on these medicinal plants, and the plant itself isn't doing a good job of killing the parasite while it's sitting there in the feces, then could it actually be that these medicinal plants are acting as kind of hubs for infection because they're harboring more of the infected poop than the uninfected or the non-medicinal plants are. So that's kind of a bit of a off topic from your question, but made me think of
Yeah,
it.
no, thank you for answering it. It's interesting about all the spillover then of diseases from other bees to other bees if it's such a popular flower. Yeah, so lots of
Yeah,
questions. So cool.
yeah, and definitely this parasite has a pretty wide host range. So, most bumblebees are infected by it, but also some solitary bees as well.
How about honeybees?
They seem to be so there's other closely related species that are more um, adapted to honeybees. Honeybees, it seems like they can maybe um, harbor it enough, like if they consume some of it, it can pass through their gut and safely be expelled the other side and still be infective, but it's not actually reproducing inside the honeybee gut.
Other folks, anybody with a question? George?
I don't have a question. I just have to say that you are doing incredible work. I had no idea that insects get sick and they can find ways they find plants that that medicate them. I had never even thought about this. That's
Yeah,
just
thanks. Yeah, I
incredible.
mean, it's pretty wild, but they're doing it. Yeah.
It's amazing
I
work
don't,
you're doing.
Well,
I
thank
don't
you.
think any of us would have thought that, George. Actually, I sure didn't. So
No,
it's been an eye opener in that regard. Something very different that we learned about here tonight. So
yeah.
you know, I I had a question about the parasite itself that you were mentioning. You said it was related to leishmania, right?
Hmm.
So what does it do to the bee, and what sort of a parasite load is going to be fatal, do you know, to the bee? Uh,
Yeah, so it seems that it's mostly what it's doing. I'd say on a physiological level, we don't really know. Like it's certainly taking nutrients from the bee. What kind of symptoms it's causing is pretty unclear. Um, in terms of what manifests in the bee, basically it makes them less efficient at foraging. So bees that are infected are not as good at handling a flower. It takes them longer to get nectar out, it takes them longer to fly to the next flower. Um, and then it's definitely more queens that are infected die over the winter. I think in general we know so little about the what's going on with bumblebees queens over winter. So definitely we don't know exactly why it is that the disease is killing them. It may just be that they go into the winter with poor nutrition because they weren't as effective at foraging as the healthy bees, but there may be other physiological effects that we don't know. In terms of the load, it's super variable, but I mean, I've seen bees where, so we typically dissect the gut out, and it's mostly the hind gut where the infection is. And so you grind up the hind gut, which is, I mean, it's tiny. Uh, and then you take a little sample of that mixed with some insect ringer solution and look at, uh, count how many cells there are in 2/10 of a microliter. So a really tiny amount. And you can find up to 600, 800 of these Clythidia cells there. So it can be really an intense infection.
so, I mean, when you think about leishmania in uh, human context, it's a horrible thing and
Mhm.
flesh eating type of stuff. But this relative isn't doing the same thing to the bees, obviously, is it?
No, yeah. I mean, it's in the same family, but it's a quite diverse group of
Right.
parasites. So there's, yeah, these uh, Cryphidia are exclusively insect hosted. There are species like there are mosquitoes that host a lot of other species of Cryphidia, but in insects, they don't seem to have any of these really awful symptoms that we see for leishmaniasis or Chagas in humans.
All right. Well, thank you. Other folks, anybody else have any comments or questions they would like to ask? It's certainly your work opens up the doors for a lot of questions, as you were saying, and it's obvious that there's a whole lot more work that could be done in many different directions. So anyway, other questions, please. Okay, well, we're getting overwhelmed with questions here.
No question, but Gordon, you're doing some really interesting stuff. My wife and I were just talking. I just told her about it. Like, we had no idea, and it makes sense. Insects can get sick, and they find ways to medicate. And who knows? What about mammals, and what about reptiles?
Yeah, no, absolutely. I think it's definitely all the animals are figuring it out.
Well, we know mammals do. There's
Amazing.
foraging behavior on mammals that indicates, medication. That's for sure.
Yeah. Yeah, thanks, George. Very nice of you
Sometimes
to
you
say.
just need some chicken soup.
My wife's a NICU nurse, and she's very intrigued by this discussion.
Ah, okay.
She takes care of the little babies.
Preemies, yeah.
Fred, when I heard Gordon's talk, I don't know, 2 or 3 years ago now, and it was just a mini talk at that point. It wasn't an extended, 1 hour talk. It was just 10, 15 minutes. And when I first heard this, I said, this is fascinating. I think this would be a great presentation to give to MES. And it was.
It certainly was, yeah.
Yeah, thanks so much for the invite.
Thank you so much for accepting. And we have some wonderful gifts for you, Gordon, on behalf of MES.
Wow. Thank you.
If you would be so kind to email me your preferred mailing address, we're going to send you a journal, our once a year Maryland entomologist journal, which you would like. It's a nice little journal. And Jean, who you see on screen, is our editor, who does a fantastic job of this journal, better than a lot of little journals that you see from different organizations. Ours is quite good, I have to say. So we'll send you that. And we also have an honorarium for you, too. So,
okay. Thanks so much.
Yes, indeed. So yeah, send me your information and we can get that stuff sent to you. You're also, by the way, a 1-year member of MES, courtesy of being a lecturer for us tonight. Okay. Awesome. So we hope that you'll join us again. And we hope that maybe we'll see you in Toronto one day. Who knows? Maybe we'll come visit.
Yeah, please. Please let me know if you come through.
It'd be cool. Yeah, but like to see what more you do with this area of research too. And maybe there's a lot more plants that you can discover that are of value of this sort. I'm sure there's got to be a lot more. So,
yeah.
All right. Well, thank you so much. Everybody can show a reaction sign there.
Thank you, Gordon.
Yeah. Thanks to all
of you. Thanks, Gordon. Go Canada.
Go Canada. All right.
Bye, everybody.
Bye, everybody.
Okay, the MES members, we'll just do a quick business meeting. So if you stay on, that'd be great. Okay. Everybody else is welcome to stay if they want to, but this is just a little bit.